Welcome everybody. Thank you, Andrea, for the introduction of design, what it is in general. And thank you, Jan, for inviting me to this beautiful place here. Today I want to talk about RNA design. So actually we are not able to design any chairs, even if they exist already. And it will be on theory and practice. So there will be a theoretical part and then a few applications. More of the practice will be during the hands-on session that follows after 1.5 hours of my talk. Then we have a coffee break, and then the hands-on session will be directly during this morning. So you know that picture already, or at least a version of that. Ivo showed it on Monday. It depicts the basic paradigm of RNA structure biology, right? So you have a sequence, it folds into a structure, and this structure determines or dictates more or less the function. So we have these 3 steps: sequence, structure, function. This is true for many of the non-coding RNAs we know about. And well, let's play a game. You see a structure, and you guess the function. What's this? Right. What's this? Someone listen to Ivo? Yes. Even that's the trickiest. But there should be an expert on that. Marburg. No. So actually you have the RNA on top. Variants of that, so that's a consensus kind of depiction. So just stick to that thing here. And you have an open promoter, as the textbook would tell you, with a -10 and a -35 box. And this RNA mimics an open promoter. Yes, it is the 6S RNA. What this guy does is it takes the polymerase when the bacteria starve, so the RNA is expressed if nutritions come rare. And you get a huge amount of this RNA in a cell, and the polymerase recognizes this motif, binds to that central bubble thingy here, and stays there until the bacteria can grow happily again, and polymerase is released. It's kind of parking lot for the polymerase. So, well, there might be many, many more structures and functions behind them. But what we're going to talk about today is the inverse. So RNA design aims on the reverse. So we want to have a structure, we want to get the sequences. We have a function in mind, we implement that as structures, and then we need the sequences that perform actually in a cell the function. Right? So there are 2 main questions I'd like to address during my talk today. The first one is, which sequence optimally folds into a given target structure or structures later on? And the second one is how to implement novel functions that might not exist yet in a cell using RNA design. But for the beginning, talk about this first question: how to generate sequences, how to optimize sequences, such that we are at least in silico sure that they adapt the structure we want to have. Ignoring structure and sampling sequences, nucleotide acid sequences, we already know or heard about that. We have 4 possible assignments on each position. So if we have a string of 12 nucleotides we want to assign, and we have no other constraints than that, we can assign to each of these positions an A and C and G or an U, right? This ends up into about 17 million possible sequences. And many of you might know there are sequence constraints, like you want to have a certain nucleotide at the second position of your RNA or the first position to get it transcribed, or there's a ribosome binding site to recruit your ribosome. Things like that are frequently encoded as sequence constraints, and if we modify this toy example up here towards a thing that has sequence constraints, our possible sequence space, our sequence space collapses to 3 million if we apply a G at the second position and the V here, where the V stands for 3 possible nucleotides to assign to that position. So it's 1 for the G, 3 for the V, and 4 to the power of 10 for the remaining ends. Well, there was this first picture, sequence, structure, function. So there was no structure so far. What about structure? So we know already from Ivo and Ronnie that RNA forms base pairs. Even random sequences form base pairs somehow. And we could have such a structure constraint, which is here shown as a dot bracket notation. I will use this representation of an RNA structure most of the time during my talk. So what is it about? You have a base pair that's an open bracket and a matching closing bracket. Many of them, all these base pairs could be assigned to one of these pairs or tuples of nucleotides, which form valid canonical base pairs, Watson-Crick base pairs. So we have 6 possibilities to assign this first position, but this will tell what we can assign down here. And we have, in this case, in this example, 4 positions that are not restricted by any structure. These are shown by these dots, so these are unpaired positions. And here we have, again, these 4 possibilities to assign them. So they are equal to an n in sequence space, and we can assign A, G, Z, and U to them. So if we multiply the possibilities we have shrinking our possible sequence space to 300,000 sequences that are all compatible to this structure. And life is not easy. We might combine both. So we want to have a certain nucleotide at first position or ribosome binding site at some place in our RNA. So we have sequence constraints plus the structure constraints. And if we do so, we have to somehow propagate the information that comes from the sequence constraint along the base pairs we have on the structure constraint. So what we have is here G, and this says what this nucleotide over here, so the second last base pair, could be. There are just 2 possibilities to pair with a G, right? So there was this GC base pair and the GU base pair. And that's why we have this 2 written here. So these are the possibilities for that specific base pair. If we have no sequence constraint, we have the flexibility of 6 possible assignments to a pair of positions. And we have 3 base pairs that are not restricted by a sequence constraint. So this is 6 to the power of 3 down here. And the first 2 numbers are simply the 3 possibilities we got from the V, so the unconstrained positions. And the remaining are the 3 ends here, here, and here, where we have 4 possibilities to assign a nucleotide. These are all independent. We multiply them, and we end up with 80,000 possible sequences for a toy example, right? And, well, we could think how to select the sequence that is good on being in that structure. And 80,000 is a lot, right? You have a huge pile of sequences, and you want to investigate them and want to select the sequence that folds best, let's say, into your target structure. What you would do, what you would calculate, estimate. Any clues from the first day or the second? Mm-hmm. Something else? So stacking energies, so you calculate the energy of the structure, right?
The probability to form another structures, so it should be minimized.
Yeah, cool. And something in between, maybe. So there was already, this is just you take the sequence, fold it, and. Sorry? You could assign that as well, right? But to do so, you have to do a very basic thing, let's say. You could also ask, does my sequence folded into the MFE form this target structure? So you have a stable, a sequence that folds stably into the target structure. And actually, I will go through all the possibilities step by step and guide you through to a way or a measure that seems to work quite well for single-state RNA design. So single-state RNA design in that case means you have just one structural constraint. And well, as mentioned from the audience, we could calculate the energy of the target structure for each sequence. That's just a reminder. So we fold the sequence into our target structure up here and ask what are the energy contributions of the individual loops, right? So exterior loop, this hairpin loop, and all the stackings along the way. And then we see already with these two sequence examples, both folded into the same structure in our into our target structure, that, yeah, well, they are different instability. So we have minus 4.8 kilocalorie per mole for this guy here, and minus 5.3 for this guy here, by simply assigning the energies. Okay, so let's have a look onto our 80,000 sequences, calculate the energy of struct for all of them. So I call this calculation just energy of struct. So given a structure and a sequence, tell me what's the energy that corresponds to that. And this is just a histogram plot. So you have the energy of struct on the x-axis and counts how many sequences have a certain energy of struct on the y-axis, which is important. What is important for all the subsequent plots I show you is that the things we want to have are on the right-hand side. For the energy of struct, I flipped the axis. So positive values here, we don't want to have sequences that have an energy of struct that is positive, since it's not stable at all, and negative value is over here. And I will do this for all the remaining plots if necessary. So whenever you want to pick a good candidate, either look on the right-hand side of such a histogram, or if there are two histograms plotted, then look at the bottom right. They are the good candidates. Okay, if you look at it, yeah, we see, well, there are a lot of different energy of struct, so it's a broad distribution, and we might favor those guys over here, right? They are stable and they can adapt our structure. But does the target structure correspond to our actual minimum free energy conformation? That's not necessarily the case, and what we simply can do is predict the MFE energy and structure for all our sequences again, right? And then plot this against our energy of struct measure. This is shown up here, so this diagram you should already know as the energy of struct thingy. And here you have a bar plot, so to say, or histogram for the minimum free energy. Again, the count is here on the y-axis. But it doesn't matter how many there are, so that's why I skipped the axis labels on the y. And if we combine those two measures, then we see there are many things that have an MFE are predicted to be 0, so the open chain, something like that. And they almost all correspond to the high EOS values, right? Something you would expect. But you also have guys where you have a reasonable stable energy of struct with -4, but there's a discrepancy to the minimum free energy, which is here -6. So these guys you might select by your EOS measure, but not by the MFE comparison. And well, you would take those guys down here or those at a diagonal. Since you know my energy of struct is the most stable structure, or my target structure is the most stable structure that this sequence can adapt. Those guys are on the diagonal. And you would prefer those ones which are more stable than the others, right? So we favor low energy of struct candidates that are equal to the MFE. But as you all heard from Ivo, there is more than the minimum free energy structure, a most stable structure. And if you look at this toy example here, or the sequence example here, you see what might happen. So the MFE structure is shown here. It has -5.3 kilocalorie. So it would be a perfect candidate if we selected on given this plot. So it's somewhere here. But if you look onto the ensemble, so taking the upper triangular of this dot plot, we see there are at least 2 alternatives shown. So, which means there's another structure that is, in this case, even equally stable. So it's also -5.3 kilocalorie, but it's different from our target structure. We have a tetra loop here and CG-rich stem closing that, and our target structure should be shifted. So we want to have a unpaired nucleotide at the 3' end and just 3 base pairs, 3 nucleotides in the loop. Well, so we know there's something still hidden on our measure or behind that, and we need to get a number on that, right? And we see from at least this example that the MFE state is not the only stable state, and in fact, it's rarely the only stable state or only stable conformation a sequence can adopt. So the structures ensemble might be really diverse. And there was this energy that somehow represents your ensemble of possible structures, which was called the ensemble free energy. And Ivo told you how to calculate that and how to really predict it in a fast way. I just give you the formula here. And what we could do with this ensemble free energy is that we can compare our minimum free energy state to the ensemble free energy and use this as a measure. But let's plot the energy of struct against the ensemble free energy. So what you would like to have is that your energy of struct is the same, or almost the same as your ensemble free energy, so that the whole ensemble is dominated by your target structure. This is what you would like to have, right? And these are these guys down here again. And if you compare that to the plot of the energy of struct versus MFE thing, then you see that the distribution gets shifted towards lower ensemble free energy candidates, and that you sort out somehow those guys over here so they get shifted slightly, so there are differences. And you would again take those on a diagonal. So we would favor those sequences where we have a low energy of structure for our target structure that is more or less equal to the energy of the ensemble. And with that, we could calculate these probabilities. One probability you heard of is the probability of a certain structure in the ensemble. So we ask for the probability of the energy of struct conformation, so to say, for our target structure. And this is simply, e to the power of the energy of the target structure minus the ensemble free energy divided by this RT value, the gas constant and temperature. Right, that's this Boltzmann weight again. And if you do so for our example where we already know there's something in the ensemble, we realize that we can calculate the probability and then we see, well, our target structure is only populated to 44% in the ensemble. And we can calculate that for this guy over here as well, and we get the same numbers. It's equally stable, right? And of course, we could also calculate that for this guy up here, this gray one. And we see it's populated to 10%. And what we would like to have is that almost all the conformations are equal, or that are populated in the ensemble should be equal to our target structure. There are people who like defects more than, for instance, the probability. So if you want to calculate the probability defect, then it's simply 1 minus the probability of your target structure in the ensemble. And what you should keep in mind is that this measure only takes the target structure as it is into account. Only this single structure is asked compared to the ensemble. And you can plot this probability defect, again, the broad distribution, and you would take those guys where the defect is minimal, so the probability of your structure in the ensemble is high. Right? But as I told you already, this probability defect or the probability of the target structure in the ensemble just takes this single target structure into account. But there might be more structures that are similar to our target structure. So maybe it is, for instance, the structure that doesn't have the outermost base pair could be also stable, populated in our ensemble, and it's not that different from our target structure. And maybe don't care or care that much that it has to be really this structure we gave at the very beginning. Some flexibility, so one base pair more or less would be okay. Then the ensemble defect is a measure that takes similar structures into account. So you add up similar structures in the ensemble which are similar to your target structure and count them as well. So you enlarge somehow your sequence space again, so the sequence space of good candidates, while you say, okay, the ensemble defect measures ensures that similar structures to our target are favored, while others that conflict with our target structure are prohibited. How can we do that? In a simple way, we could just go through our structure ensemble, so generate all structures your sequence can fold into using, for instance, RNA subopt. Calculate the probability of these structures in the ensemble, and then multiply this value by the distance to your target structure. So we have a structure, we see a structure, P, and we want to know how far we are off with that structure from our target structure. If this distance is large, we want to get a high value. If this distance is small, we want to get a low value. So the defect should be low if all the ensemble is dominated by structures that are more or less the same as our target structure. And it should be high if the ensemble is diverse and there are many, many other structures. And the base pair distance used here, just for simplicity, is how many base pairs do I have to open in one structure and close in order to get the other structure, to transform P to our target structure. And if we do so and we plot all that, so up here is the probability defect, the best measure we had so far, right? Take high probability candidates, and that's it. And here is this ensemble defect. So 0 is again down here, so the best candidates are down here at the right bottom. Then we see, if we calculate an R-square of both distributions, so to say, of the then we see the R-square is fairly high, so we have 0.8 of correlation, so to say. So both values correlate, but they are not perfectly the same. And what you see here is you have candidates in your sequence space that have a probability or probability defect that is around 0.8, but the ensemble defect tells you all the remaining structures which are here, which you miss with the probability defect, are similar to your target structure and should be counted. So you get an ensemble defect that is pretty low, close to 0. Right? So we would take those guys down here. With that, we know, or this guided you through the simple thing, designing or selecting a sequence that is able or most likely folds into the target structure you want to have. And what we can learn from this list over here is, or we made some notes during all these plots and all the things we were investigating so far, and we realized, okay, there are two important conditions that have to be fulfilled. One is we want to have the target structure to be thermodynamically a stable structure. Right? This was what we had at the very beginning. So we want to have our target structure to be the MFE, so to say. This is what people call the positive design. Or if you want to solve that, it's a positive design problem. On the other hand, we learned by investigating this probability defect and the ensemble defect that we would like to reject or prohibit contrary structures, so that conflicting with our target structure. And this we did with the ensemble defect, and rejecting these contradicting structures is known as a negative design goal. So positive, you want to make it stable. Negative, you want to get rid of alternative structures in the ensemble in the best way. And there are many programs that all address the positive design problem and to some extent the negative design problem. And the list is by far not complete, so there are still coming new software out that uses genetic algorithms or Monte Carlo simulations or whatsoever. And they all try to address this problem. They sample sequences in a different way. So not as we did here, generating all of them. Since if your sequence space is larger than our toy example of 80,000, you just have to sample and look how well the taken sample is, or how good the taken sample is. So maybe you want to also guide the selection of the sampling process towards stable structures. This is what incarnation does. Here you do a Boltzmann-weighted sampling, so you get from the very beginning already sequences that are likely or that fulfill this positive design. So you take sequences with a high chance of which the MFE structure is the target structure you want to have. And then you have subsequently to optimize for this negative design goal. But I don't want to go into details what these tools are all about. They, at the end, solve the same problem or address the same problem. And here I like to quote Richard Dawes, because he says, well, you could come up with fancy ideas in theory, they might be brilliant, but at the end, if you really want to take a sequence and implement it in a cell or in a tube, if you go to in vitro, then nature scores, right? It doesn't matter how you get these sequences. You will probe a handful or maybe 2 handful, and you want to have functional RNAs designed. And one example of a nice study where they showed that you can succeed with RNA design is the one by Alexander Green from 2014, where they designed or where they solved the inverse folding problem for 5' UTR. So they said, well, we want to have 5' UTR where the ribosome binding site and the start codon are somehow covered in loops of a stable structure. The idea is that if the RBS is in a loop and the AUG is in a loop, on the one hand, that the ribosome can't access them, so the ribosome is not able to open up this structure and start translation. That's one thing. And the other thing why they put them into the loops is that you do not get sequence constraints on your design problem for the base pairing part. So you have the complete flexibility for these pairing positions here. So you get rid of these sequence constraints in these regions, which shrinks your search space, right? And this was not done before, so putting them in a loop. And people might say, well, if the ribosome binding site is in the loop and it's really like the ribosome scans and looks for this short 6 nucleotide motif, then it has to find that and bind there and open up that structure, right? The ribosome is such a huge thing, this little structure can't compete that. But in fact, it can. If you look down here to this flow cytometry data, you see that the switch alone, so this 5' UTR in front of a GFP reporter shows autofluorescence of the cells. So it's pretty off. Autofluorescence means these cells emit green light, even though there's no GFP, no protein encoded on a plasmid or in the genome. They do just because they can. And we want to add more GFP, more green light, so to say. We want to enlighten them. And with this off state, let's call it the off state, it should be similar to the autofluorescence, ideally. Something which is maybe hard to achieve. And you can only achieve it if you make this structure really stable and dominating the ensemble. How they did it is they used NUPAC design, one of the tools I've shown you, and the guys from NUPAC, they invented or they described for this ensemble defect and used it for optimization. So they showed it is really possible to switch off translation by a structure similar to that. And then they add a trigger RNA, so just an RNA sequence that is able to bind to the 5' end of this switch RNA. And by that opens up this structure, so it gets linear more or less. And the ribosome should be able to access now the ribosome binding site and start translation down to the reporter gene. So what we would like to have if the trigger is there. So if you have switch and trigger, you want to get high values for GFP fluorescence. And they're impressively high in this study. So more than 100 fold change they achieved, which is great or impressive. So they really tweaked their reporter system such that such high fold change values are possible. So comparable studies, they have a fold change of 10 or something like that for the best candidates, just to give you a rough measure how well the reporter system is tweaked to show this impressive data here. And what they did is they used an iterative design approach. So they had a first round of designs just using NUPAC and deciding on several parameters like loop size and things like that, and toehold size. So how large has to be the complementary region that is unpaired at the 5' end in order to get this trigger RNA binding. Things like that. So they varied that, they investigated, do we see any correlation on that? Can we improve our toehold switches? And they investigated also by in silico screening. So they really took the system apart and showed that they're able to regulate gene expression just by solving the reverse folding problem for a single RNA using the ensemble defect. So with that, I would say, or I hope I convinced you, that it is possible to solve the inverse folding problem for just one structure and to get reasonably good candidates using the ensemble defect, right? Of course, there are always results or things that are still hidden. So there's this nice game approach by Richard Das, where people just play and try to solve the inverse folding problem without any, or supposed to be, without any algorithm behind. So they just assign nucleotides, see what's the structure, so they really probe the structure and learn somehow how to assign nucleotides to a certain target structure such that the probing experiment, like the shape experiment Ronnie was talking about, tells you, yes, you achieved the goal to assign the correct nucleotides to get your target structure. And there they say, or they show that they can even outcompete this ensemble defect measure for some instances of target structures. And this suggests that there are more than the ensemble defect you could take into account given complex and diverse set of target structures. So I tell you, yeah, the ensemble defect is fairly good in doing the job we want. So getting a target structure and the sequence to the target structure, but there might be more hidden in nature. And as we all know, or as I said before, Richu always says nature scores and not the computer, right? So with that, I'm done with the first part. So the single target structure design problem solved, so to say. And I would like to continue with the multi-structure design problem. So we have now not just one structure that we want to have. We want to have two structures, two alternative structures. As you've seen in the example before in the dot plot, now we want to select for sequences that are able to switch, so to say, or to populate two different structures. And well, let's do it the same way as we did it before. It's maybe pretty simple, right? We have two structures and a sequence constraint. We know that structure tells what we can assign to another precision, so a base pair. This G here says what we can assign down here, right? The pairing partner of a G has to be C or U. And let's do that. So follow these brackets along our two structures. So the first position is a G. We know the first position is paired in both structures, and we can assign a sequence constraint to the corresponding positions over here. So this G is somehow telling that the Y, so a C or U has to be assigned here. So this Y stands for these two nucleotides, C and U. And it tells also, following the first structure constraint that the second last position has to be a Y as well. And now we could just continue and propagate this information. So assigning the Y here tells you the second position has to be. So we have a C or U at one side. We have the possibility of A and G on the other side, right? And that's shown as an R here. And then you continue. You follow simply the base pair, the base pairs within each structure. And at the end, you see that the structure constraints changed your sequence constraint, right? And what you could do now is simply, well, take this sequence constraint, generate all the compatible sequences. And if we do so, we shrink our search space from the one target structure example. So you hopefully realize that the first structure is still the same as in our one structure example. Shrink down from 80,000 to 12,000 possible sequences. Why? Because the first position is a G, so it's fixed. Then there are 8 positions where we have either an R or an Y, so 2 possibilities to assign. And we have 2 positions with an N. They are flexible. So to say, we can assign all nucleotides we want to them. So 4 possible assignments to these 2 positions here. And we have still this V in our sequence constraint, which was equal to 3 possibilities of nucleotides, right? And yeah.
Okay, sorry, because here I don't know why I'm taking the first nucleotide to be J. Because I think it's not the only possibility to have a base pair between the first and the last nucleotide.
Yeah, but at the beginning, we started with that, right? So it was a sequence constraint that the first position has to be a G. It's set from the very beginning. So there was some reason why we took that. And then we propagate this information through using the structure information and end up with this sequence constraint, so to say. So what we did, we propagated individual constraints, so the structure constraints and this first G to multiple positions of our sequence. And, well, ended up with this number of possible sequences, right? Okay, but can we do better? We know that sequence constraint alone didn't help that much when we looked at the one target structure example, right? We could also just take the structure constraints and ask how many sequences are compatible to these 2 structures. And how can we do that? An easy or a fairly easy way to depict this is you draw your nucleotides here shown as the position, so 1 around the clock till 12. In opposite order than the clock. The first position, so first nucleotide, second, third, and so forth. And the last nucleotide is shown here and corresponds to that last N in the sequence constraint here, which is actually no constraint since all assignments are possible. And then you take the first structure and you connect those positions with a straight line that are paired. For our blue structure up here, we would draw a straight line from the first position to the second last one, and from the second position to the third last one, and so on. And if you do so, we end up with a plot like that. So we have the RNA backbone around the circle and base pairs connecting individual positions shown here as blue straight lines. And on top of that, we could have the second target structure you want to have. We put it the same way. So we connect the first position with the last, the second with the second last, and so on. And what we end up with is such a zigzag path through our RNA sequence. So the first nucleotide is connected to the last one and to the 11th position and thereby to the second and so forth. So what we want to know is how many possibilities do we have if we have no sequence constraint to assign nucleotides or base pairs to these positions. And let's do it for a base pair, just to have an easy starting point. If we want to know how many possible assignments do we have for a base pair, we simply enumerate them, list them, and know, well, there was, at the very beginning of my talk, possibility of 6. So 6 base pairs, and we know how many there are, but there should be a more general way to calculate that or to know that. And this is hidden, or can be described as a base pair table, shown here. BPT means base pair table. And what it's encoding is on each column an A for the first column, C for the second, G, U for nucleotides, and the same for the rows. So the first row is A, the second row is C, third row G, and the last row is in U. And you have a 1 in this matrix if the nucleotide encoded on the row pairs with the nucleotide encoded on the column. So we have A in the first row and U in the last column, so we have a U pair encoded here. Same down here, the third row corresponds to G. So a G can base pair with a C, which is encoded in the second column, and with a U, which is encoded in the last column. So it's simply alphabetic order of A, C, G, and U, which you should imagine on top and on the side of this matrix. And if we have this base pair table, we can say, okay, let's sum up all the ones because they correspond to a base pair, right? And then we end up with this information of 6 possible base pairs. Okay, that's maybe much of work, but what for? Well, we can extend this and, for instance, ask how many possible assignments do we have for a path of length 2. So 2 base pairs that depend on each other, that overlap at a certain position in our sequence. And of course, we could again enumerate all them. So we have a base pair that's simply the same list as before if we want to know how many base pairs we have or possible base pairs we have. But this time we extend this by another base pair. So the first position is paired with the second position in this really toy example, and the second position should be paired with the third position, right? And by that you get combinatorics. So if you have an AU pair, you have an U at that position, it's clear what you could assign to the third position. Either G to form a UG base pair or an A. Right? So we have 2 alternatives encoded if you assigned an AU to the first base pair. Same for CG, so you could pair the G with an U or with an C. And you can go down here, and at the end, you will realize, well, there are 10 possibilities for having dependency of 2 base pairs. And, well, enumerating them is not that efficient, right? And we have the base pair table up here already. Maybe you can use that as a tool to estimate these 10 possibilities. And yes, we can. So we have a path of length 2. We simply multiply the base pair table with itself. And then we end up with this matrix. And this matrix actually encodes for an AU base pair that we have 2 possibilities to assign the second or the last position in our path. For AU base pair, we have to look here. No. yes, for AU, we have to check here, and we see there's a 2 encoded on our base parent table. Well, now we know if we have just one possibility, like if we have a GC base pair, there's no alternative way to assign the third position. It has to be a G. Then we have a 1 here, and if we have 2 or more possibilities, we have a 2 in here. And we can again sum up over all the elements of our matrix, and we get the correct number of possible assignments for a path of length 2. And as you all might already guess, if we want to know how many possible assignments we could make for a path of length L, it's simply multiplying the base pair table L times with itself, and then summing up all the entries.
So do you know a closed form for that?
Well, not from the top of my head.
So this is the Fibonacci number of n plus 2.
Yeah, right. And if we go back to our example shown up here, we know there are 3 positions, those guys here, that are not connected to our path, to our red-blue path up here. So we have full flexibility to assign a nucleotide there, times the length of this path, so times the base pair probability matrix multiplied 8 times, and summing up all the values in there. So doing so, you have 178 equal possibilities to assign sequences to that red-blue path, and this ends up in 100,000 possible sequences that are compatible to these 2 structural constraints. Right? So now we solved the structures constraint problem. Sorry?
64 multiplied by 178 is doesn't equal.
I made a mistake.
It's like 6,000 should be.
Yeah.
About 6,000.
Okay. Yeah. I see. Well, we shrinked our search space, right? Sorry for that mistake in the slides, I corrected. But what we actually want is, we want to have both. We want to have sequence constraints and structure constraints fulfilled in our design problem. And how can we do that? Well, we can again draw our dependency graph, so to say. So we overlay both structures we want to have. And this time we have not just the position here, we have the nucleotides assigned to positions, or the sequence constraint assigned to the positions. Up here we have a G, so the first position should be a G in our sequence constraint, as we said it before. And we have a V down here, right? But this we don't care of that much because it's an unpaired position, so that's not going into our structure evaluation. And the reason why I highlighted this G here is that it's a special point along the path. And we call this thingy articulation point. We call that articulation point or concatenation point, something like that, because we have to separate our path at such points. We cut the first base pair out, so we have a problem to assign sequences to that part and then to the remaining of the path. Given at the first position at G. This we didn't have so far as well. And what we could do is, well, we know this path has a length of 7. So we use our base pair table again, multiply it 7 times with itself, and end up with something like that. And as we know, we have a G at the first position. We not simply add up all the entries of our matrix. We only add up the entries in our matrix that are in the row that corresponds to a G nucleotide. So the third position in this matrix, the third row in the matrix. And there we have 34 possibilities. So this path can have 34 different sequences. Then we have two possibilities up here. So we have a G paired with something. It's again a C or U. So we have in orange fulfilled this base pair out here. With 2 times this 44 we calculated from the path given the first position is a G. And these 2 black numbers here correspond again to the unconstrained positions in our sequence constraint. And then we end up with 3,000, right? Well, but what is this all for? So we count sequences like hell, and why? We just could assign nucleotides and base pairs and see what happens. And if we do so, we get already problems if we try to assign or sample a base pair. So imagine you have a base pair that pairs x1, so position 1 with position 2. And you draw along this decision tree, which is shown here, in a uniform way, which way to take. So you add a question mark on top, and you ask what to assign to the first position. And you take 1/4 for A, 1/4 for C, G, and U. So 25% for each of the possible assignments for the first position. And then you do the same, depending on where you are, for the next decision. For base pair, it's the last decision. So if we end up in an A, then it's pretty clear we have to assign a U to the second position. If we end up in U, we have to assign an A or an G to the second position, right? And if we do that, then we realize uniform decision is not uniform sampling. So we would like to get all the base pairs equally likely. So there's no reason why we should have an AU base pair sampled more often than a CG base pair, right? And if we follow the decision tree, we see at the first decision we take 1/4 for A times 1 for the U, and then we realize, okay, with 1/4 we take an AU base pair. Compared to a UG base pair, which we take 1/8. So 25% and 12%. It's not much, but it's already enough. And if you extend it to longer paths and more complex structures, it's not what we want, right? We want to sample at the beginning, or we want to see at least with some probability all the sequences in our solution space. And well, what we could do is we know how many possibilities we have after assigning the first decision. So we know that we have 6 possible base pairs. We want to assign in the simple example 6 base pairs combinations, nucleotide combinations equally likely. So we could weight our decision, our first decision, compared to the possibilities we will have after that. And this can be written in formulas and is shown up here on the decision tree. So we weight the decision to take an A at the first position with 1/6. So because there's only one nucleotide, one pair assignment encoded behind that decision, right? You can't get just one base pair if you assign an A at the first position. And you can get 2 base pairs if you assign an U to the first position. Well, So that's why we would like to know the number of possible assignments for a certain path or certain dependency in our dependency graph. And that's why we want to calculate these numbers I've shown you before, to weight our decisions along the decision tree. And well, you might think, well, that's already a lot, but yeah, okay, let's do it that way. I understood, hopefully, how I assign sequences to paths, to base pairs, to unconstrained positions, things like that. That's still okay, but if you increase your toy example to a more real-world example, and if you increase the number of structures you want to fulfill as a constraint, then your dependency graph, which is shown down here, gets also more complex. So you become, these are the nucleotides of that example, so 35 nucleotides are spread here, and if one position depends on another one, then you have these lines between them, right? You get these graphs. And for this small example here with 3 structures, you get circles, you get paths. So this corresponds to a single base pair. This corresponds to a circle. If you walk around here, you end up at the end at the starting position. That is also possible. And if we increase the number of structures, states we want to have, we get rocket structures in our dependency graph. And if we increase further with 5 structures, we get even some strange animals in our dependency graph. And we have to take care to assign the sequences which are possible for that animal in a correct way. And we decided at the first step to do it in a uniform way. And we all solved that and wrote a program called RNA Blueprint that is able to sample sequences compatible to multiple structural and sequence constraints. And it's drawing all the possible sequences fairly in a uniform way. And of course, if you want to publish something, you have to show that you're somehow better than the others, or that the others might get problems in the way they do their sampling. And we show that with this graph in brief. If you have a bias in sampling, so you see certain sequences over and over again, you slow down the process of exploring the sequence space. So if you take AU over and over again, it might take long until you see a GC base pair, so to say. And this becomes, of course, more complex for larger problems, multiple structural constraints in sequences. So this is all on GitHub, and you're going to use it today during the hands-on session. And, well, this solves the sequence sampling problem. There are other methods out there, like RNA redprint, developed by Stefan, Sebastian, and Jan, where they again sample sequences that are likely to be stable, or that form stable structures, where its target structures are stable structures in the ensemble. So you can introduce biases if you want to have them, right? So you want to sample low MFE structures, for instance, and you can put that on top. But if you do not uniformly sample and you see stuff over again, you introduce biases you might not be aware of. So you might get sequences, or it is possible that you don't see sequences you would like to have, basically. So you can extend this RNA blueprint approach, do it for RNA much better, so to say. But let's stick to this RNA blueprint tool. Well, this solves the sampling problem. What we didn't have till now is an objective function, something like the ensemble defect we had for the one structural state problem. So I've shown you the ensemble defect for just one target structure seems to be a reasonably good measurement, right? And well, that's hard to get for multiple states because you want to implement the switching or depend on temperature, depend on a ligand, depend on another RNA, things like that. So you have two conformations that you somehow have to weight within your ensemble, and that's more complex than getting one structure and a sequence that folds into that structure. Of course, there are ways to describe a certain goal, and this example here, this objective function shown here for this two-target structure, for our two-target structure example, is somehow, or it's more or less the equivalent for having the probability defect converted to two states. So the first term just tells we want to have both energy of structs of both structures, both energies of these structures to be dominating the whole ensemble, right? So we say in the one target case, the energy of our target structure should be equal, ideally, to the ensemble free energy. Then we have just one structure. Here we have two structures, and if you add them up, they should dominate the ensemble. They should populate 100% of the ensemble. And then, of course, we have to somehow decide, should they be equally stable? Should there be a difference between the energies of these two structures? And here we decided, well, just keep it equally stable. And you can come up with many, many, many other objective functions depending on what you want to have. And, well, given an objective function, you can score sequences, and you see they, again, perform differently. And it depends really on the goal or the application you would like to implement, which objective function you need or you would like to apply. And from case to case, you have to develop your own objective function. There's no general objective function you could say, that's the thing you always want to have. And that makes it also hard, or yeah, again, you can come up with fancy methods and do benchmarking and show that you're much faster or whatsoever than the others, but nature scores again. And here nature has to score two structures and is switching, for instance. So the problem becomes more complicated. And that's the point where I like to come to my last part, or to the last part of my talk. It is how to implement novel functions, how we can do that. And I just want to show you one example. And if we start, or if people start to design RNA sequences, they typically follow a common design pipeline, which has 4 major steps. The first step is you take your biological system where you want to test your device in, or your structure, or the thing you designed, and you have to analyze that. And you should know it by heart, ideally. Then you somehow sample and optimize your sequences. So you have to come up with an idea what you want to implement. Then you come up with an objective function, and you sample sequences, and you optimize the sequences. So you select those that best fulfill your objective. After that, typically people do an in silico analysis, which could be, for instance, calculating kinetics for all the candidate sequences you generate is too expensive. So doing keyfold trajectories for thousands of sequences might be not the good idea. So you postpone such analysis and do it after selecting sequences that presumably that fulfill your objective, and you want to see, is the kinetics the right one I want to have? So you, again, filter the good candidates using this in silico analysis, and then you go for biological testing. And I want to follow this pipeline while showing you one example which we're currently working on, and it is interfering with the process of transcription and translation. So hopefully all of you are familiar with a plot like that or with graphics like that. So we have the DNA helix, we have the polymerase sitting on it, producing an RNA strand, in this case producing an mRNA strand. The ribosome gets to know about this mRNA, it binds to it, and it translates the message to a protein, right? And the thing, when we want to somehow play around or interfere with this process, and the point where we want to do it is the tRNA. So we want to get a hands on the tRNAs, and especially their maturation. What does this mean? Maybe it's hard to see, but down here you have a pre-tRNA, so if it's transcribed, it has on its 5' end a leader sequence, and on its 3' end a 3-nucleotide motif called CCA. So there's always the CCA encoded, or typically encoded in a genome, and they come out like that. And what is necessary to get this tRNA working and this translation process is you have to load it with the amino acid, and you have to get rid of this leader sequence, and the CCA is needed as recognition site to also plug this amino acid to it. So to mount the amino acid onto the tRNA. And after this processing step, so RNA-seq is recognizing there's a tRNA that's not ready yet for work, I have to process it. Then, if it's processed, it can bind to a certain message, depending on the codon-anticodon, matching, and translate, or helps to translate this message. And well, as I told you, you should know your biological system by heart, or you need a way to get a readout if your RNA is processed or not, right? And here also this readout mechanism is shown. So we have the LAX-Z gene encoded on the chromosomal DNA, but it has an AMBO mutation, which means there's a triplet in that can't be translated by natural tRNA. We need a special tRNA that matches this triplet and then can help to really translate the full message. And if the full message is translated, we get beta-galactosidase activity as a readout. So we can screen for that. Well, how to interfere with this maturation step? Ivo already told you that I'm working on riboswitches and on riboswitch design. And what we did in the past is we used an aptamer sequence selected by Selex as a starting point. So the specific aptamer sequence shown here is able to sense theophylline as a ligand. And we turned this aptamer, shown here in red, into a switch by attaching sequences to it. On the one hand, a spacer sequence and a perfect complementary part to the 3' end of this aptamer. And by that, we can switch into that state. We add several U nucleotides behind that. So we have a stable hairpin plus 8 uracil nucleotides following it. It's a perfect intrinsic terminator for E. coli, for instance. So this thing should terminate transcription. And if theophylline is bound, the red part is stabilized, and we do not get such a terminator hairpin and can continue transcription. The thing is active, so if you have a reporter gene behind that, we see green light, And we have a switching from an off to an on state in that case. In the very first study, we succeeded with a four-fold increase of translation. So we know how to design these things, and we know how to test them. But well, just plug it together, right? Take a tRNA, plug it onto this ribo switch, maybe get rid of this poly-U stretch, because we don't want to terminate anymore. We really want to switch between accessibility of a leader and sequestering this leader structure in our hairpin. And if it is sequestered, so covered, then RNA-SP, shown here as Pac-Man, shouldn't be able to process the tRNA, so shouldn't be able to cut this leader sequence. And if we bind theophylline, we want to have this leader accessible. The RNA-SP should recognize it and process it. So the tRNA should be able to swim around and do its job. The poor man's approach is to simply copy and paste sequences. So our collaborators did it and just tried. So you have part of the switch here, and then you attach the tRNA to it. And you cross fingers that something happens. And well, yeah, something happens. You get a fold change of 1.7, but that's not much, right? Even if you can reproduce it six times. It should be better. There should be a way to do it better. And when we got these results, we started investigating these results, and we thought, well, there is a difference between designing a terminator hairpin or transcription termination switch and an tRNA RNA-SP switch. And it's the timing, actually. If you have a transcription regulating ribo switch, you need a perfect timing that during transcription, if no theophylline is present, you have to form this hairpin rather fast, such that polymerase is really kicked off at that site. And if this is not formed sufficiently fast and at the right time point, polymerase just continues transcription. It doesn't care that there might be at one time point in the life of the RNA a stable hairpin. So if it's gone through, that's it, right? And so we have here time dependence, depending on the transcription speed. And here, we do not have such a constraint. We have a switch. We have the time that we need to really transcribe the whole thing, since we need a complete tRNA for a working device, right? So we have to transcribe the whole switch. And then it has to go somewhere, or the RNA-SP has to recognize this molecule and has to cut it. So the time we have is much longer. So getting to equilibrium or close to that is much more likely in that case than in that case. So that's a difference, right? And we said, well, we learned from many other studies that plug and play is not the way. Unfortunately, it's more like plug and pray, right? So you stick sequence together and hope that it's working, and we want to do it better, and that's why we really did the in silico stuff, sampling and objective function here. So these two states, we want to have one state, the leader is sequestered, and in the other state, in the bound state, we want to have the leader accessible. That's basically it, right? We can use, again, RNA blueprint to sample sequences that are compatible with these two structures and the sequence constraints hidden behind them. And then we can come up with complex, but maybe complex only, objective functions that describe our system, right? And to put it simple, we want to have a high probability of getting something like that, so of getting the leader accessible if we add theophylline to our system. And we don't want to see the leader if no theophylline is in the system. And this is more or less written here. So the probability of being the leader accessible in the ensemble given theophylline, so as a hard constraint or soft constraint, implemented. This one reflects that over here. This should go to 1, right? You want to have this. If theophylline is present, you want to have an accessible leader, it should be cut. And this here in the back corresponds to that state. So we don't want to have the leader accessible if no theophylline is present. That's why we take the 1 minus this probability here. Of course, we can describe that system in much more detail, which we did down here. But essentially, let's assume it does the same due to time constraints. Right? It's more complex. It more precisely describes the system, but in a way it does its job. So we optimized sequences, sampled sequences, took the 5 best ones. After an additional in silico analysis, I skipped here, and we went to lab, and our collaborators did the experiment, and they tested and saw, whoa, at least 2 out of 5 are functional, and the one of it is really switching with a good fold change. So 35 fold more beta-galactosidase activity if we add theophylline. That's kind of a success story. Well, still there are candidates that should work, that if calculated, these objectives are perfect, but they don't. So there's something going on we are not aware of right now. We are investigating that. We have ideas what's going on there, but this is always the case. So you go back, you analyze your system, you go back, you try to improve your design, you do the experiment, and so and this in a circle, ideally, but this rarely happens in real world, unfortunately. So we can't make our design tools or our objectives better if we don't get this feedback from nature, so to say. Yeah, with this slide, I just want to advertise the hands-on session that will follow the coffee break, where you're gonna design a ligand switch, this time without any transcription translation regulation, just for simplicity. And we will also have a challenge where you somehow change this design to a temperature switch, or that's the challenge you should. We hope you get able to do that during this hands-on session. With that, I want to thank lab members that work on RNA design and our administrative staff in Leipzig, the bioinformatics group, all the collaborators, especially those guys from Leipzig who worked with us on this RNA-SP switch design, and of course all the others down here for their collaboration and ideas. With that, I want to thank you for your attention, and I'm open for questions.
Back to the way of enumerating the sequences and counting the number of sequences, why don't we use the fact that, for example, the GC base pairs with 3 H bonds are supposed to be more stable than AU to maximize the stability of the bonds of the helixes? Why don't you introduce bias in this way?
That's exactly what this RNA red print does in a way. So what you want to have is you want to get the energy model into this sampling process, right? In a way. And one thing you could do is you say, I weight my decisions by the energy I get if I take a GC base pair, for instance. Right? So GC is more stable than AU. You can do that, but at that point you neglect that you, in the nearest neighbor model, you have stackings. So what you have to take into account is additionally the backbone. So what's the next decision you do, and then you have the energy of a stacked pair, things like that. And that's becoming more complicated. And in our framework, it's hard to achieve since we don't know, or we don't take care about the backbone while assigning the individual components of the dependency graph. But in principle, it's possible, and you can guide the sampling process towards stable structures. If you want to know more about it, then ask this guy over there. This time it happens to you, right? Yeah. Ivo, next question is yours.
Do you have to additionally optimize your sequences, you know, to be like in the normal GC content for E. coli, or maybe, you know, because you sample random sequences, your sequences can be very out of equilibrium for normal sequences in the cell?
True. There are approaches out there that really take care during the sampling that you get a certain GC content. One for this single state design, the first one, or one that took it into account, was Incarnation from Jan. And there was even another tool. It's using an ant colony optimization algorithm to draw these sequences, and there they showed they can precisely design a GC content. Yes, you can do that. For sure, it's a good idea if you want to have your RNA working in a certain species with a certain GC content, things like that. But I think yeah, you can do that, but it's not the overall goal. You can add this later on as well. So you can do it during the optimization, right? You add another term and then you get a GC content as well. And I think it's not necessary to get 35.0 GC content.
So I have a small selfish question. There's a general belief in the community that some of the shortcomings of manual design, or even the efficacy of some of the in silico design, is due to the existence of kinetic traps. And actually GC content has a rationale in this context, because you might not create this kind of spurious helices. Mm-hmm. Is that something you've experimented with on the experimental side, or is that something you believe, first of all?
No, so we didn't go in that direction so far and played around with that. Since for our designs, we have, so the last two things I've shown you, we have an aptamers given for the transcription termination thingy. You have the aptamers given, you have a perfect complement to that aptamer 3' end. So if the aptamer is not transcribed, or the GC content or whatever doesn't fit to your system, then this aptamer won't work, right? And it's dictated what's in the 3' part, so getting the perfect complement. So the only real flexibility we have in there is this loop region, right? And well, things might went wrong there, but not necessarily if you have just 10 nucleotides, right? It's something you have to keep in mind when you see there are devices that are not functional, right? That's true. And also for this RNA-SP switch, we have just a little flexibility where we really design.
Is it possible to design a piece of sequence to be an aptamer for some random molecule?
Yeah, at the first evening, this question came up several times, and people are interested in that. Honestly, there are the CELEX experiments, right? Where you have a ligand and you select an RNA sequence from a random pool, or RNA sequences from a random pool that are able to bind to your ligand of interest. And it's pretty hard to get a working sequence from that experiment already. So there's a handful of aptamers that you could use in a cell as a sensor. So there's not much how you could learn how to design these sequences. And it is pretty hard to get. In principle, you could select for any ligand an RNA sequence using the CELEX experiments, but only in principle. So there are many attempts where people failed to really get a sequence that binds specifically to your ligand. That's a big issue. And for these RNA aptamers, you rarely, or what you want to have is, you want to have something like theophylline, where you have this rod structure, the ligand binds, and then it's getting compacted. So we have free nucleotide pairs, so to say. So we have triple helices in there, and it's getting really compact and packs the ligand. There are other examples where this might not work, at least if you look on the secondary structure. It can't pack the ligand. It can't recognize that precisely or that specifically the ligand. And that's also a big issue here. So you have to cover the ligand. You have to see all the sides of your ligand to really get it specific. And that's pretty hard. I think there might be ways to do it with 3D structures and MD simulations, things like that. You might get into that direction, but I'm far from being an expert on that, and I can't imagine that it works for a completely new ligand. For something where you have already an aptamer, you can somehow guess what to look for, right?
So in the interest of the coffee break, we'll stop here, and thank once again Sven.